30. This is the beginning of recorded lecture 1-5. We are going to start and finish chapter 4, cell structure. So this slide should look familiar. I introduced the concepts of prokaryotic cells and eukaryotic cells way back in the very first lecture of the course. Of course, these are not to scale. Remember, prokaryotes are like 1/10 the size of eukaryotes. I just have it blown up so we can more easily compare them. And so in a chapter about cell structure, of course, we have to talk about the different structures that these cells have. Instead of comparing and contrasting every little thing that these two cell types have, the easier way to go about this is to talk about the stuff that all cells have, because there are plenty of things that prokaryotes and eukaryotes have in common. Then, once we've introduced those, we could talk about the more complicated things that eukaryotes have, because of course you remember prokaryotes, small, simple, single-celled organisms, not a lot of stuff going on, basically bacteria. Eukaryotes, much bigger, more complicated, a lot of fun stuff on the inside. This is us. This is plants and animals and fungi. So, okay, let's get into these cells, the structures that all cells have. Let's start with a plasma membrane. So this, of course, should be familiar. We talked about this a little bit in the previous chapter. All cells have this lipid bilayer. We can see it labeled here. It's just sort of a thin line in the prokaryotic cell, an even thinner line in the eukaryotic cell. But yeah, a plasma membrane is something that all cells have. Another one is cytoplasm. This one's defined in the key terms as the entire region between the plasma membrane and the nuclear envelope. More about the nuclear envelope in a couple of minutes, but basically the cytoplasm is the inside of the cell. It's just mostly water. It's just the stuff that everything is floating around in. Cytoplasm is not labeled on the eukaryotic cell just because it's already a pretty busy figure, but yeah, the cytoplasm is here. It's just all this stuff around the organelles. It's what they're floating in. It's mostly water. Yeah, it's all the stuff on the inside is the cytoplasm. Another thing that all cells have is DNA. You can clearly see this in the prokaryotic cell here, DNA. It's less clear in the eukaryotic cell. It's housed within this structure called the nucleus. We'll talk about that in a couple of minutes, but there's DNA in here. All cells have DNA in one place or another. Also, ribosomes. These are structures that if we look at them on a sort of zoomed out scale, they look kind of like red dots here. Again, they're so small in the eukaryotic cell, they're not labeled in this. Oh, here they are, ribosomes. Tiny little dots you can't even really see. If you were to zoom in on them, we'll get into this detail in a later chapter. These are protein factories. These are structures that build proteins. Obviously a very important job for the cell, so it makes sense that there would be a lot of these. So to talk a little bit more about ribosomes, they build proteins. They're made of RNA and protein. So this is a callback to a concept from the previous lecture when we were talking about DNA and RNA. And I said that RNA was a short-term copy of DNA. Usually it's just information. But I did mention there were, even though most RNAs are just a message, they're data, they're instructions, I did mention there are some RNAs out there that have structural roles. And here we go. This is one of them. There is RNA in the structure of the ribosome that is required for it to do its job. Build proteins. And of course, there are many of these per cell, and they're things that all cells have, prokaryotes and eukaryotes. So that does it for the things that all cells have. Now we can talk about all the extra fun stuff that eukaryotic cells have, because in addition to all those things, eukaryotic cells have membrane-bound organelles. So the cell itself has a membrane around it. These organelles we're about to introduce have their own membranes. And, I've been using this figure as a generic eukaryotic cell. I'm going to use this figure for the rest of this chapter. This is showing a sort of typical plant eukaryotic cell and a typical animal eukaryotic cell. There are a few things that are plant only, and there's one thing that's animal only. They're color-coded for your convenience here, but I'll bring these up as we come to them. This is going to be sort of our main figure that we'll return to as we talk about all these crazy things inside of these eukaryotic cells. So we'll start by talking about membrane-bound organelles with the nucleus. Yeah, I've mentioned this a couple of times already. This is usually the biggest, visually most obvious thing inside of most eukaryotic cells. It's this big purple thing in the center of the animal cell, this big purple thing. I say it's purple. They're always drawn purple. It's because when you stain these things, the most common stains when you're looking at cells under a microscope, they show up as either blue or purple. So that's why all the drawings color code them like that. But yeah, here we're seeing the outline of the cells in green in this fluorescent stain microscope image. And yeah, look at that nucleus. It takes up like a third of the entire cytoplasm, a third of the entire cell. This is a really big structure. So what does it do? Well, the nucleus stores DNA. Again, prokaryotes just have DNA floating around in the cytoplasm. The nucleus keeps this safe. And like I've shown, this is typically the biggest, most prominent organelle within eukaryotic cells. If we want to zoom in on this structure a little bit, here we go. These are just a couple of different images from different textbooks. This one's flat, this one's more 3D looking. There's a lot of labeled stuff in here that you don't need to worry about, but something I do want to call your attention to is the membrane. Again, this is a membrane-bound organelle, and it's not just one membrane, it's actually two. The nucleus is a very important organelle. DNA is very important. It's the instructions for how the cell is supposed to go about doing everything. It's kind of like the brain of the cell, tells the cell what to do. And so you want to keep the stuff inside very safe. So there are going to be an inner membrane and outer membrane that compose what's called the nuclear envelope. Now, we do want to keep the stuff inside secure, but there need to be ways for things to go into the nucleus and to go out of the nucleus. So this is a very effective barrier, this nuclear envelope, but there are also structures called nuclear pores that carefully control things going in or coming out. So the nuclear envelope actually is in the key terms. Let's read what the key terms document says for this. It is a double membrane structure that constitutes the nucleus's outermost portion. And nuclear pores control entry and exit. Now, DNA is something we saw just in the last chapter. You know, this DNA double helix should look familiar. Eukaryotic cells have a lot of DNA. I mean, a lot of this stuff. Giant double helix. There's so much DNA inside of your cells, inside of any eukaryotic cell. It could not fit in the nucleus. It couldn't even fit in the entire cell if we had it sort of naked like this. So we do not have, by we I mean eukaryotes, we do not have naked DNA double helices. What we have is DNA packed up. Again, this is done so that it can fit within the nucleus. Again, a couple of different figures just from different textbooks. The DNA double helix is wrapped around these proteins. They're coiled around one another, they're coiled around again, and they form these dense structures called chromatin. And here it is again, the DNA double helix wrapping around these proteins called histones, coiling up again, and we refer to this as chromatin. So chromatin, if you want a more formal definition, the key terms define it as a protein DNA complex that serves as the chromosome's building material. So yeah, that's how we package our DNA. Next membrane-bound organelle, we come to a structure called mitochondria. So back to our overview, we can see mitochondria, these orange-looking pills here. They're in plant cells, they're in animal cells. If we want to zoom in on these a bit, here's a bigger view of mitochondria. A lot of labeled stuff here that you don't need to worry about. What does this do? What is the purpose of this organelle. Again, in general, organelles all exist to fulfill some specialized function. Well, the job of the mitochondria is to break stuff down, to break down organic molecules like sugars and generate something called ATP. We're going to talk a lot more about ATP in a couple of chapters, so you'll get your fill of that then. For now, this is just an energy storage molecule. This is how the cell fuels its chemical reactions. This is what the cell uses for energy. So we're breaking down stored energy and generating usable energy. That's the purpose of this structure, to do those chemical reactions. Now, as it turns out, a lot of those chemical reactions, a lot of the enzymes that are doing this breakdown are located in the membrane. So we see, just like the nucleus, this structure also has an outer membrane and an inner membrane. And these very important enzymes that are actually doing the job here are in this inner membrane. So you'll notice, whether it's a microscope image here that's been colorized or, a cartoon drawing, that the inner membrane has all these folds sort of going up and down, up and down. These are folds within the membrane. These structures exist in order to have more membrane surface area. Remember, the important enzymes are embedded in the membrane. If this was just a flat oval, you would be able to cram in a certain amount of those enzymes, but by having all this extra area, you can cram in way more of these important enzymes. So many of the chemical reactions are in this inner membrane, and the folds of this inner membrane exist to increase surface area so we can have more reactions. Oh yeah, there's also something weird about these mitochondria. You may have noticed on those labels. They have their own DNA and their own ribosomes. So we can see, DNA labeled here, these little circles, and ribosomes, the little dots here. And that's weird, right? We just said that the DNA is stored in the nucleus, so why does this organelle have its own DNA? Well, this is a story actually for BISC132, much later. The quick version is these mitochondria are thought to have once been free-living bacteria that got into a eukaryotic cell and have been there for so long, they're now codependent on one another. These things cannot live on their own as independent living organisms, and the eukaryotic cell can't really live without them either. The reactions that they're doing in this inner membrane are that important. However, they do retain some amount of independence by having their own DNA and ribosomes. So you don't need to worry about the backstory. I think it's cool. But the important thing to note is that these organelles do, strangely, have their own little amount of DNA and their own little protein factories, their own ribosomes. While we're on the topic of breakdown, there's a couple other organelles that are involved in breaking stuff down. One of these is called the peroxisome. So, unfortunately, peroxisomes are not pictured. I really like this overview. They're not listed here. If you wanted to know what they look like, they're boring. They're little circles within this eukaryotic cell. Here's a cool microscope image of crystallized protein is why we're seeing this diamond shape here. So what are these boring little spheres doing within the cell? Well, within the peroxisome, we are breaking down lipids and fatty acids. Sound familiar? It's this stuff, so stuff like, triglycerides. So this does kind of beg the question of, why don't we just do this in the mitochondria? The whole point of the mitochondria was to break down organic molecules, and this is definitely an organic molecule. Why do we have a peroxisome? Well, without getting into the biochemistry of it, there's something that happens when you break down fatty acids. This is unavoidable. Anytime you're breaking down this type of organic molecule, you are going to generate a byproduct. That byproduct is called peroxide. Now, if the word peroxide sounds familiar to you, there's a reason for it. It is the ingredient of hydrogen peroxide, this brown bottle. And if you know anything about hydrogen peroxide, yeah, antiseptic. This is what you put on, a scrape or a cut or something in order to, prevent infection. The reason why this prevents infection is because this hydrogen peroxide is incredibly deadly. It kills stuff. It's very dangerous to not just our cells, but to any bacteria or other microorganisms that might be getting into that cut or scrape trying to cause a skin infection. So hydrogen peroxide, or peroxides in general, are nasty, dangerous things. And when you're breaking down lipids, you're generating this nasty, dangerous chemical inside of your cells. And that's a liability. We don't want this causing havoc on all the other stuff within our cell. So that's why we have peroxisomes for lipid and fatty acid breakdown, so we could generate this byproduct and safely, in a secure way, neutralize it. So to summarize, we need an organelle for this type of breakdown because lipid breakdown makes harmful hydrogen peroxide. The purpose of the peroxisome is to contain and break down that peroxide byproduct. That's why we don't just do this in the mitochondria. There's a third organelle involved in breakdown. It is called the lysosome. We can see lysosome on our big picture here. There's one labeled in the, again, it's just kind of a boring looking circle. There's one labeled in the animal cell. There are lysosomes in plant cells. Again, when we get to things that are plant only or animal only, I will call those out. There's not one shown in this plant cell, but it is found within plant cells as well. So what's the deal with the lysosome? Again, it kind of just looks like a boring, plain-looking sphere. Well, the lysosome has a low pH inside of this boring-looking sphere. And low pH denatures proteins. So this is really good at making sure we can break down proteins. Additionally, there are enzymes within this lysosome, the little crystals shown here, that could break apart those proteins once they have been denatured. So here's the lysosome in action. Here's a cell taking up some big food particles, sending them to the lysosome. The proteins within that food are going to be denatured and broken down into smaller pieces. But that's not actually the only thing the lysosome is capable of doing. The lysosome is also capable of dealing with old or damaged organelles. Here's a familiar looking, although kind of screwed up, mitochondrion. Something's wrong with this thing. You send it to the lysosome, it's going to break that entire thing down. And in fact, it doesn't have to even be large internal structures. The lysosome can deal with external stuff as well. There are many cells in your immune system that have lysosomes that will deal with invading bacteria by eating them, bringing them into the cell, and then sending these pathogens, these bacteria, to the lysosome to be broken down for parts. So really, this is just an all-purpose scrapyard for dealing with large structures and with proteins, denaturing these things and neutralizing things. All-purpose scrapyard is a great way to think about this. So how do I summarize? Lysosome. There's a low pH inside that denatures proteins, and there are digestive enzymes that are built to work at low pH that can break down proteins and large food particles and foreign material and or damaged organelles. It does all of this stuff. Let's talk now about vesicles and vacuoles. So, well, what are these things here? Well, there's a vacuole labeled, a little unimpressive looking sphere here, a bigger looking sphere in the plant cell. Vesicle is not actually labeled on these things because they're much, much smaller. What do they do? Well, vesicles and vacuoles are both responsible for storing and or transporting materials. The difference between vesicles and vacuoles is that vacuoles are bigger than vesicles. That's why vacuole was labeled here. There's no label for vesicle because they're tiny. We'll see them in other figures. They're going to be involved in other steps and other stuff within the cell. But yeah, it's just like a vacuole, but smaller. They store things, they transport things. They're found in both plants and animals. However, there is a special type of vacuole called a central vacuole that is our first plant-only thing. We could see it labeled here. This just says vacuole, but this is a central vacuole. It is bigger than the nucleus within plant cells that have it. Its job is to store water. Again, that's something that plants are going to need to do photosynthesis. Animals don't do photosynthesis, so we don't need this large central vacuole. So it makes sense that we would both have vacuoles, but the central vacuole thing is plant only. Speaking of plant only, that's a good segue into the next organelle, chloroplasts. We can see it labeled in green here, chloroplast. It's the green-looking oval, green-looking pill here. If we zoom in on the chloroplast, yeah, there's a lot of labeled stuff going on, but the job of this chloroplast overall is to do photosynthesis. That's why it's plant only. That's why it's not found within animal cells. Now, we'll talk way more about photosynthesis in a couple of chapters, and what we will see is a lot of the chemical reactions for photosynthesis take place in a membrane, embedded within a membrane. Does this sound familiar to you? It's exactly what I said when I was talking about mitochondria, that a lot of these chemical reactions take place in a membrane. And in order to maximize the amount of reactions, that's why these mitochondria had these folds to cram in even more membrane. Well, this is true for chloroplasts as well. In order to cram in even more of these enzymes, they don't have those same folds, they have discs of membrane. They kind of look like, poker chips or Oreo cookies or whatever. This is actually inner membrane that's been folded up into these stacks. And these weird looking structures exist in order to have more of these important enzymes. So, same thing, copy and paste, exact same thing I wrote for mitochondria. Many of the chemical reactions of these chloroplasts take place in the inner membrane. Folds increase surface area for more reactions. Oops, did I accidentally copy and paste this too? Well, nope, it was no accident. Just like mitochondria, chloroplasts, even though it's not labeled here unfortunately, chloroplasts do also have their own DNA and ribosomes, and the story is exactly the same as it was for mitochondria. These things used to be free-living photosynthetic bacteria, swallowed up by some ancestral eukaryotic cell, and now they're just an organelle, but they have some remains of their independence by having a little bit of their own DNA, and they make their own ribosome. So this is true for chloroplasts as well. On to a big one now, the endomembrane system. So this is actually three things, or there are going to be three components to this, but they all fit together into an overall system. We can see the endomembrane system labeled here. It's this weird stuff around the nucleus and this blue thing right here, as well as some of these other dots are associated with it as well. And yeah, it's found in plant cells, also found within animal cells. So yep, there's this stuff around the nucleus and this blue stuff off to the side here. If we want to focus in on this, because this is a little busy looking, if we want to focus just on the endomembrane system, again, a couple of different figures just from different textbooks. We could see this one in particular shows us all three components color-coded, the blue, the green, the orange. Again, they're not actually these colors. That's just convenient to look at. But what all three of these components have in common, they are made of folded sheets of membrane. So it looks just like, a line or something like that. This is a lipid bilayer. So the stuff inside of this endomembrane system is kept separate from the cytoplasm. So to summarize that, there are three components to the endomembrane system. They're all made of folded sheets of membrane. And yeah, this system as a whole is attached to the nuclear envelope. We kind of see this here, how it's associated with the nucleus, and we can see it in the big overview here, how these folded membrane sheets are associated with the nucleus. So what does this actually do? Because it is pretty big, it is pretty impressive looking. The overall function of the endomembrane system is to modify and transport proteins. So we talked about ribosomes building proteins. And yeah, a ribosome can, make a protein, connect the amino acids together, build this protein. But for eukaryotes, a lot of our proteins are pretty complicated. They're not just a string of amino acids. A lot of our proteins need to have extra stuff done to them. They need to have tags added or pieces removed and rearranged, or have carbohydrates attached to the amino acids or lipids attached to the amino acids. So it may seem unnecessary. The ribosome can build the protein. What do you need this for? Well, this is going to modify those proteins and ship them to where they need to go. So we'll see this process by looking at these three components. The first of which, the one in blue in these figures, is called the rough endoplasmic reticulum, or ER is endoplasmic reticulum, the rough ER. It's called rough because even under a microscope, a strong enough microscope, you'll see that this folded sheets of membrane are studded with ribosomes. That's what these little red dots are here. So yes, ribosomes are involved in this process. Before we can modify and transport proteins, we do have to build proteins. So the ribosomes are building proteins and then sort of threading them into this system as they're being built. So yeah, proteins are made and inserted into the system. There are some modifications done to the proteins within the rough ER, but hold on to that thought. Most of the modifications are going to be done later on in the system. So that's the rough ER. What about this green one here? I don't know why this textbook doesn't even show it. The next component here is called the smooth endoplasmic reticulum, or the smooth ER. It's smooth because there are no ribosomes, which tells you there's nothing with protein being made here. No ribosomes means no protein. Instead, the smooth ER is where carbohydrates and lipids are made. So we're going to see carbohydrates and lipids involved later in this system. Again, they're going to be attached to our proteins as a modification. And so, yeah, this is where those things are made, in the smooth ER. Now, I'm bringing up the smooth ER in the context of this system, one of these three components. But I should point out the smooth ER has a side hustle in breaking stuff down. So in addition to making carbohydrates and lipids, the smooth ER breaks down some foreign compounds, things that need specialized enzymes, things that are a little too weird or complex or specialized for the peroxisome or the lysosome or the mitochondria to break down. So they also do this, and that is important. Once these proteins have been made and these lipids have been made, they're sent from the ERs, rough and smooth, via vesicles. Oh, I told you we would see vesicles here. And yeah, we see this labeled transport vesicle. This is, again, a small version of a vacuole used to transport this stuff from one part of the cell to the other. So the products of the smooth and rough ERs are sent in vesicles to the third component, the Golgi apparatus or Golgi body. So they're sent via vesicles to Golgi apparatus. This is where we get most of our protein modifications. Again, we don't really see this on a figure like this, but these proteins are having lipids attached to them, or they're having carbohydrates attached to them, or they're being cut and glued back together. Weird, complicated stuff that eukaryotes need to do. And once they've been modified, they're then packaged into vesicles once again, and then sent out to wherever they need to go. So this is a packaging and distribution system. Go back to this overview here. the eukaryotic cell is a complicated place. You're building proteins, but some of these proteins need to go to the lysosome. Some of these proteins need to go to the mitochondria. Some of these proteins need to go to the nucleus. Some of these proteins need to go outside of the cell. So a big part of the endomembrane system is to, once these proteins have been built and modified, put them into vesicles and send them specifically where they need to go. In both of these examples, it looks like these are proteins that are destined to go outside of the cell to do some sort of job here. But yeah, this vesicle coming off of the Golgi apparatus could go to any place within the cell. So yeah, shipment is a very important part of this process. And I do need to point out, there are lots of proteins we make that only function when they've been modified properly. Prokaryotes don't need to do this, but for us, for eukaryotes, we need this complicated system for a lot of our proteins to get properly functioning proteins. Eukaryotic things we've been talking about were membrane-bound organelles. The endomembrane system and chloroplasts and mitochondria, all those things had membrane. However, there are other not membrane-bound structures that are specific to eukaryotes that we need to talk about. So the first one I want to introduce is something called the centrosome. We can see the centrosome on our big overview here. This is labeled centrioles. It's the same thing as we'll see in just a second. However, it is labeled in red. This is our first animal-only structure. Chloroplasts were plant-only. The central vacuole was plant-only, but the centrioles or centrosome are animal-only. If we zoom in on what this structure looks like, we can see, yeah, there's one centriole, there's a second centriole. That's why this is sometimes called centrioles, but the term centrosome refers to both of these together. The purpose of this structure is to organize microtubules. That's these little small tubes. That's what microtubule means. These orange small tubes here. These are going to play an important role in cell division. And the purpose of this structure is just to keep these organized. Why is this animal only? Don't plant cells need microtubules as well? Well, they do. They do cell division. They have microtubules, but this organizing structure here, it is in fact animal only. Another important not membrane-bound structure is the cell wall. So, a plant-only structure. So the purpose of the cell wall is to provide protection and support. So we can see the cell membrane. We see cytoplasm, then cell membrane, then cell wall. So this does not replace the plasma membrane or cell membrane. It's outside of the plasma membrane. It is protecting the cell from the outside, sort of like an outer shell. I should point out, this is actually found within prokaryotes as well. They build it out of different compounds, but yeah, prokaryotes in general have a cell wall also. The purpose, rigid external protection and support. Made of various polysaccharides. Call back to this slide. When we were talking about polysaccharides, I specifically used cellulose as an example. That's part of the plant cell wall here. So, yep, that's fun to come back to. These cell walls are made of polysaccharides. And yeah, I don't want to say I don't want to say plant only because they're within plants and within prokaryotes. So I'm going to say the cell wall is not in animal cells. But don't animal cells need protection and support, external protection and support? Well, we do. We just don't use a cell wall. Instead of a cell wall, animal cells use something called the extracellular matrix, or ECM, because that's a rough one to say. We can see, well, we can't see the extracellular matrix pictured here, but if we zoom in a bit, we can see it labeled here. There's the plasma membrane, there's the cytoplasm, and all this junk on the outside is the extracellular matrix. Instead of being a very thick, rigid wall of polysaccharides. The extracellular matrix is a loose amalgamation of proteins and carbohydrates just sort of stuck together. So this is a good protective barrier, but it's not as much of a shell as the cell wall of plants. Sorry, I didn't bring this up earlier, but yeah, here's a thin line of plasma membrane. Yeah, that's how thick the plant cell wall is compared to the lipid bilayer. So this extracellular matrix is not as strong, but it's a lot more flexible. Animal cells in general need to be able to squish around and move a lot more than plant cells. So it makes sense that their external protection would be not quite so rigid. So the extracellular matrix, flexible external protection and support. It's made of proteins and carbohydrates, and yep, this one is animal only. So the cell wall and the ECM were external protection and support. But it's also important that we have internal protection and support. So this is, and something else, is carried out by a structure called the cytoskeleton. Once again, this is not pictured on my big overview here, but if we zoom in at a cross-section of the cell, we can see the cytoskeleton kind of everywhere, right? We see a familiar mitochondrion. We see a familiar looking rough ER. The cytoskeleton is all this other stuff. These orange filaments, the green filaments, these red filaments. There's a lot of empty space, it looks like, on these big overviews in between these organelles. There's a lot of not anything pictured here. In reality, there's not a lot of empty space within the cell. All this space in between one organelle and the next is connected by all these different cytoskeletal fibers. So this cytoskeleton really connects everything in the cytoplasm. And there are 3 major types of fibers that make up the cytoskeleton. We'll number them because why not? First one up is microfilaments, also known as actin filaments. They're color-coded in red here. So I'd take a step back and talk about this name, cytoskeleton. So when we talk about a skeleton, we probably think of our own skeleton, which is very, very durable. It's inside of our body, but it doesn't really move around. You know, you've got joints and hinges and stuff like that, but your bones are not changing their shape. That's completely the opposite way with the cytoskeleton. That's where the analogy kind of breaks down. The cytoskeleton is a dynamic structure. Unlike our bones, this stuff dissolves and reforms itself as the cell sees fit. We can see this by looking at the actin filaments in action. Don't sweat all this alphabet soup here. This is from an immunology paper, and immunology always has a lot of this kind of stuff. But we see the little red filaments here building actin in order for the cell to change its shape and reach out and grab this target. So this is a dynamic structure. Oh, I pulled up this figure at the beginning of this chapter in order to show you the nucleus in blue, but guess what's labeled in green? It's actin. We're able to see the entire outline of this cell by labeling actin because actin, just like the rest of the cytoskeleton, is everywhere within this cell. So actin filaments are involved in changing the cell's shape. They're also involved in muscle contraction. There's actin labeled here, and that's a topic for another course. But yeah, I should point it out at least a little bit that these actin filaments allow the cell to change their shape, and they are involved in muscle contraction. Filament number 2, intermediate filaments. They're called intermediate because they are intermediate in size. They are thicker than actin filaments, but they're smaller than the third type that we'll get to in just a minute. The job of intermediate filaments is a lot more straightforward. They exist to be strong. Again, the cytoskeleton is internal protection and support. Intermediate filaments just give strength to these cells. Cells chock full of intermediate filaments include hair, include fingernails, include horns. For example, these structures have their strength because of these proteins. So yeah, intermediate filaments provide structural stability. I think that's all I need to say about them. The third one is microtubules. And hey, this should sound familiar. We saw these being organized by the centrosome and those two centrioles. We zoom in here. Yep, that's a microtubule. It's a small tube. They're involved in a couple of important things. One of these is cell division. And again, we got a whole chapter on cell division coming up later in the quarter. We'll see this action in this exact slide later. But yeah, microtubules are important in cell division. They're also important in the movement of vesicles. So we didn't really see this. If I could back to our slide on the endomembrane system, when we had vesicles going from one location in the cell to the other, it kind of looked here like it was just magically floating from one place to the other, just like, whoop, here we've arrived at our destination. That's not actually how it works. The way vesicles get from one place to the other is by riding the rails. And that's what these microtubules are within the cell. They're a railway system for vesicles to be transported using these motor proteins in order to get from one place to another. So, in summary, microtubules have a key role in cell division, and microtubules serve as the transport rails for these vesicles to get from one place to another. Finally, last two, other non-membrane bound structures, flagella and cilia. So I'll show these in just a second, but I do want to point out these are optional structures. All eukaryotic cells are going to have, microtubules and intermediate filaments and so on, but flagella and cilia are truly optional. Some cells have one but not the other. Some cells have both. Some cells have neither. They're optional. Once again, they're not shown in the overview, but here are some fun cells that do have these. Flagella and cilia are both extensions from the cell, sort of sticking outward. Flagella are long whip-like extensions. There's usually only one or sometimes a couple of these on cells that have them, and they whip back and forth and allow these cells to swim around. Cilia also beat back and forth, but because they're so much shorter, in order to actually help the cell swim, there are usually lots and lots and lots of cilia on these cells that use them. So as you saw, these were highlighted yellow, meaning they're defined in the key terms. The key terms definition for flagella is a long hair-like structure that extends from the plasma membrane to move the cell. Definition of cilia, short hair-like structures that extend from the plasma membrane in large numbers and function to move an entire cell or move substances along the cell's outer surface. So they're both extensions, they're both hair-like. You could have long hair, you could have short hair. The flagella are long, the cilia are short. There are many cilia, there are few flagella. Now, this does bring us to the end of this chapter, and oh boy, this has been a lot of stuff. So I want to point out that in my slides on Canvas, there are a couple of tables here that summarize everything that we've gone through. Now, you don't need to know all of this. Some of these we did skip, like the nucleolus. But, if you want a table like this, again, it's in my slides on Canvas, kind of just summarizing what we've talked about. But really, this is no new information. Everything that I wanted to say, I've already said as we've gone through these slides. I just wanted to point out that these exist in order to keep things organized. So that does bring us to the end of this chapter and to the end of Lecture 1.5.